In this video, we will discuss the renaturation in detail. First, let's compare the denaturation methods. We have three types of methods: heating method, alkaline sodium hydroxide method, and salt induced method. Salt combined with acids, heat, or alcohol. The highly fast, highly accurate, and precise method is the sodium hydroxide method. It takes less than a minute. And the reversible methods are heating method and sodium hydroxide method. Heating method is reversible by cooling, while sodium hydroxide method is reversible by using phosphate buffer. While salt-induced method is highly irreversible, it disrupts the DNA structure permanently, and it is used when bulk of DNA processing is required, for example in DNA extraction and DNA precipitation techniques. While in high precision lab protocols we use sodium hydroxide method, alkaline method, and in PCR the most commonly used method is the heating method. Now let's discuss the DNA renaturation. Renaturation is the rejoining of single-stranded DNA into double helix. It is also known as reannealing or hybridization. As you can see here, upon heat, the double-stranded DNA will begin to unwound, and upon more heat, the double-stranded DNA is converted into single-stranded DNA. And on cooling, the single-stranded DNA will rejoin again into a double helix. This is known as renaturation, reannealing, or hybridization. Now, renaturation occurs by two processes, nucleation and zippering. First, nucleation will occur, that is, short complementary regions on both strands find each other. The complementary regions will join together, and then zippering occurs. The strands align and rapidly pair up. As you can see here, this is the area of nucleation where the complementary regions uh, join together, and then once nucleation has occurred, the renaturation is rapid due to zippering, and zippering will occur and double-stranded DNA will be formed. This is a cyclic process, as when the temperature increases, the bases will unstack again, hydrogen bonds will break. As you can see, the DNA is becoming partially unwound due to adenine and thymine base pairs will begin to separate because they are less stable due to two hydrogen bonds. And then if the temperature is increased, the GC base pairs will also get disrupted and we will get two single strands. Now, if the temperature becomes low, the complementary regions on both strands will rejoin again to continue renaturation. Now, the typical conditions required for renaturation include a moderate to high salt concentration. As I have discussed in the denaturation video, that high salt concentration alone will causes renaturation. High salt concentration contains cations in high amount, and cations will shield the negative charges on the DNA. As we know that the phosphate backbone on DNA is negatively charged, and the single-stranded DNA are highly negatively charged due to this phosphate backbone, and these single strands will repel each other. So, a high salt concentration will contain high positive cations, positive ions, which will neutralize the negative charge on the phosphate backbone. So, no longer repulsion will take place and the two strands will join together. Means this will neutralize the two strands. Phosphate buffer is used, which will neutralize the pH, maintains a stable pH, and the temperature, that is 20 to 25 degrees Celsius below the melting temperature, allows the base pairs to reform slowly and accurately. The melting temperature of DNA is that temperature at which 50% of DNA strand is denatured or converted into single strand. So we have to take 20 to 25 degrees Celsius below this melting temperature. So these are the typical conditions for renaturation. Applications of renaturation include DNA RNA hybridization, in which we match the complementary DNA and RNA strands together for identifying specific sequences on both. Southern blotting. We have discussed southern blotting in the denaturation video. That is, Southern blotting involves both denaturation and renaturation. First, denaturation will separate the strand, then the both strands will undergo electrophoresis and we will get the DNA fragments that are transferred to a membrane. And then fluorescent or radioactive labeled DNA probe is added. The DNA probe will undergo hybridization or renaturation with the DNA fragment. That is, the DNA probe sequence will bind to the target sequence on the DNA fragment. They both will join together through renaturation. So that is Southern blotting. In microarray analysis, The DNA probe is attached to the microarray surface, a slide or a chip, and analyze how these probes interact with the sample DNA. In both cases, the DNA probe interaction with the sample DNA sequence is detected. That is, the probe sequence will rejoin with the target sequence on the sample DNA. Then what's the difference between these two methods? The basic difference is Southern blotting is used to analyze one sequence at a time, but microarray analysis is used to analyze thousands of sequences simultaneously. The next is DNA repair studies. It is used to understand the DNA healing process. So these are the applications of renaturation.